Hi, my name is Ayesha Tariq and I am working in Dr. Piant Kwiska's lab at Kent State University. This presentation will be looking at the effects of reovirus infection on the editing of host transcriptome. Reoviruses are double-stranded RNA viruses and they can infect a wide range of hosts including plants, animals, and insects. We know that viral infection activates RNA editing by ADARs as a part of the innate immune response, and we also know that this regulation of RNA by ADARs is very important for normal neurodevelopment and immune function. A previous report has shown that reovirus infection does not affect the overall editing landscape, including major known targets in the host. But these results were based on only a handful of sites. Therefore, to look at a transcriptome wide editing changes, we used publicly available RNA-seq bioproject data from murine fibroblasts infected with wild type mammalian reovirus and an interferon sensitive mutant reovirus strain. A to G and T2C sites were filtered for quality and analyzed under two considerations. First, rates of editing, and then significant involvement in GAG pathways. First, we looked at the expression of ADARs across each sample, and as represented in figure 4, expression of ADAR1 was significantly high in infected samples, with no significant change in the expression of ADAR2 and ADAR3. How does this expression translates into the editing changes were looked at next. Editing rates were first examined broadly by comparing the overall editing sites in each samples and then narrowly for shared and unique sites among infection and control. Figure 5, 6, and 7 show that number of editing sites as well as editing rate across all samples is higher in infection, and there are almost equal number of A to G and T2C substitutions. And there were 231 sites mapped to 17 genes common across all samples, and 323 sites mapped to 71 genes that were common in mutant infection and wild type infected samples. These changes are shown in figure 7. Figure 8 and 9 show that there is no significant change in editing rate of common sites between infection and control, or mutant reovirus and wild type infection, only except a few. But you can also see unique edited sites in mutant and wild type infected samples that are significantly enriched in GAG pathways with notable enrichment in immune signaling pathways. So what does this all tell us? First, the expression of ADAR1 correlates with the number of edited sites, but there is no obvious pattern regarding rates of editing, which seems to be increasing for some sites and is decreasing for other sites. Reovirus infection elicits unique but nuanced editing changes in the host transcriptome, and despite a low viral load, the infection can still activate ADAR expression and consequently edit host transcriptome, as we have seen in the case of mutant reovirus infection, which is an interferon sensitive strain. Since reoviruses are desirable agents for virus-based cancer therapy, future studies should look at the functional consequences of editing changes caused by interferon sensitive mutant reovirus strains. This study is limited by the relatively low depth of publicly available RNA-seq data, something that could be improved in the future. Future studies should also focus on the comparison of editing patterns with related viral families such as rotavirus, which could shed light on the possible evolution of reoviruses as priority pathogens. Thank you.